Hi, I'm Michael Ralph, and today we're going to be doing a gram stain procedure. This is a particular type of staining that's going to involve everything you see right here, including all of your gram staining stains. You'll need a Bunsen burner for flame, both deionized water and ethanol, something to rinse into, and of course a microscope slide. One safety note: ethanol is very, very flammable, so ethanol and fire should never be close together. Your fire and ethanol need to always be separated by a significant distance. This is very flammable, it can be very dangerous. Your first step is to grab a microscope slide, and you need to add a drop of water to it. Just one drop. The smaller the drop, the better. But odds are good you'll get a little too much on the slide. So, grab a paper towel and just draw a little bit of water off of there until you just have a fairly small drop. The next step is to get some of your sample. To get some sample, grab a regular paper clip, and we're going to straighten it out into an inoculation loop. So that's all the way straight except for the interior loop, which is closed. We need to sterilize this loop, which we're going to do with ethanol. So give it a good rinse in ethanol over the sink. This is what's sterilizing it. Once it's done, you need to dry it off. So walk over to where the flame is with only your loop and run it through the flame once to burn off the extra ethanol. The sterilization is in the ethanol rinse, not the burn, so you don't need to cook it in the fire. Just one run through to burn off any excess ethanol. Now it's sterile and it needs to go immediately into wherever you're going to draw your sample from. Draw up some sample in your loop, and then inoculate your slide. Don't be shy. Get a whole bunch of sample into that water, and then this can immediately be flamed again to be sterilized and put away. Now we need to dry off our slide. So we're going to flame it. So walk it back over to where your flame is, and to flame a slide, you're going to run it through the flame about 2 or 3 times. Flaming a slide is not up high so that it doesn't get enough heat, and we're not cooking it over a campfire. Don't let it sit in the flame. It's just 1, 2, 3, and then let it sit. We need to dry off all of the water on this slide, so it needs to be sitting until it's dry. Don't wipe it off, just let it sit. You can flame it every once in a while, try and hurry it along, and then let it continue to dry. Once it's been sitting long enough under the flame, it's going to be completely dry. Once it's dried off, we need to heat fix it. That means now that it's completely dry, we need to flame it one more time. So that's one more pass through the flame, and that's going to stick all of our sample to the glass so that it doesn't rinse off during our staining process. Once it's been heat fixed, we're ready to actually start staining. The first stain in a gram stain is our crystal violet, and it needs to sit for 1 minute. So we'll put some onto our slide, and it sits for 1 minute. And then it's time to rinse. We're rinsing with regular water, and when you rinse your slides, remember to spray above where the sample is and let the water run down over the sample to get the stain off. If you spray it directly on your sample, you may rinse off too much stain. You may rinse off some of your sample itself. So run along the top until it's rinsed off. You will still see some stain attached to the actual sample on your slide, but most of the loose stain needs to be rinsed off. The next step is your mordant, and that's going to be some iodine solution. Again, this needs to sit for 1 minute. So, 1 drop for 1 minute. We wait. Now that a minute has passed, we rinse again. The same procedure. And now, what is usually the most difficult step is the ethanol rinse. This is where, again, we're going to take our ethanol, and after having rinsed the iodine off with water, we're going to rinse our sample in ethanol. This needs to be very fast. This is the most sensitive step. If you rinse too fast, then you can rinse off not enough stain and you'll get a false positive. If you rinse for too long, You could rinse off stain that shouldn't be rinsed off and you'll get a false negative. This takes some practice. The ethanol is a very quick rinse. Don't be shy, get plenty of ethanol on there, but then once it's all gone, it needs to be immediately rinsed with water again. That's about the speed that it needs to run at. Again, this takes some practice, but don't be shy. You need practice doing it the right way. So be fast and be thorough. After the ethanol rinse, we have our final stain, which is our safranin. It's the pink stain. Drop on there for 1 minute. Let it sit. And then rinse with the water. After your water rinse, you're ready to finally visualize your slide. So, take your paper towel and wipe off the bottom. Flame your slide one more time. Flame your slide one more time to dry off the top. And last but not least, don't forget to label your slide. Put some masking tape across the top. Don't forget to dog-ear it. And then with a Sharpie, remember your initials, the name of the sample, and the date. This is a finished slide that's ready to go under the microscope and take a look. Good luck.